Christina McNerney from the Johnston Lab at Johns Hopkins University. Today I will be showing some of my recent work trying to understand how thyroid hormone signaling drives cone subtype specification in human retinal organoids. We are interested in understanding the development of the retina, the vision-detecting tissue in the eye. Photoreceptors are the light-detecting cells. Humans have three subtypes of cone photoreceptors, blue, green, and red, which allow for us to see in color vision. How these cells differentiate properly in humans is only beginning to be understood. In order to observe and manipulate human retinal tissue throughout developmental time, we utilize human retinal organoids. Human retinal organoids are derived from stem cells. We aggregate them and then replicate the developmental signals that would be received in vivo to push them to retinal fate. As you can see, these organoids can be cultured long-term. They recapitulate human photoreceptor development as we have shown previously. In the organoids, we showed that blue cones are specified prior to green-red cones, which are too similar to distinguish with antibodies. This confirmed that there is a temporal aspect to cone specification with blue preceding green-red. Previous studies in model organisms showed that thyroid hormone receptor beta drives green-red cone fate and suppresses blue. The role of THR-beta was unknown in humans. Using CRISPR to knock out THR-beta in organoids, there is loss of green-red cones compared to the mix in wild type. THR-beta is a receptor for thyroid hormone signaling. Addition of T3, which is active thyroid hormone, to wild-type organoids resulted in an organoid with all green-red cones, and treatment of the THR-beta knockout organoids with T3 resulted in a phenotype like the THR-beta knockout with only blue cones. This showed that T3 signaling to THR-beta drives green-red cone fate and suppresses blue cone fate in the human retina. My work recently has been trying to understand how thyroid hormone is regulated intrinsically in the human retina. My first steps have been to observe in which cell types these proteins are located in human retinal organoids. We know that there are two proteins that control active thyroid hormone levels. Dio3 degrades thyroid hormone and was found to be highly expressed in early retinal development prior to cone specification. I found that Dio3 is expressed in early retinal cells. Here I have a day 92, which is young, retinal organoid stained for Dio3 in red, actin in green, and a marker for mitotic retinal precursor cells in blue. As you can see, there are both mitotic and non-mitotic cells that express Dio2. I'm currently working on identifying the exact cells that are expressing Dio3 at this early time to maintain low thyroid hormone signaling in the retina. In the process of trying to understand the expression of Dio3 in the early retina, I wanted to establish a timeline for differentiation of retinal precursor cells, or RPCs, in the organoids. I stained organoids over time and quantified the density of mitotic cells at each time point. As expected, I can see a decrease in RPC density as we progress over developmental time due to differentiation of RPCs into the different cells of the retina. This timeline is still ongoing. Dio2 is also an important regulator of thyroid hormone signaling. It converts inactive thyroid hormone into active thyroid hormone, which is T3. We know that it is expressed in late development, and the onset of expression coincides with the switch from blue to green-red cone fate. I stained a 175-day-old organoid with Dio2 in red and marked S cones in blue and green-red cones in green. As you can see, Dio2 is expressed in blue and green-red cones and another cell type that is unmarked here. I'm working now to establish what the third Dio2 expressing cell type is and repeat this stain at several time points. Another potential point of regulation of thyroid hormone signaling is the entry of thyroid hormone into its target cells. Thyroid hormone must be transported into the cell. One of the main transporters in neural cells is MCT8. Here I looked for MCT8 expression at both early and mid-retinal development. In white in the top image, I have performed RNA fish against nascent transcript. Almost all cells are expressing MCT8 at this time. Much later, at day 142, I stained organoids for MCT8 protein and saw almost ubiquitous expression. Thus, while MCT8 may be playing a role in this process, it looks like it is not unique to the onset of green-red cone specification. This brings me to my current working model. At early development, there is a large pool of retinal precursor cells. As they begin to become fate-restricted, some of these early cells express Dio3, which degrades thyroid hormone and prevents precocious signaling through THR-beta. As development progresses, several events occur. First, fewer early retinal cells are present, and so Dio3 expression decreases, gradually increasing local thyroid hormone levels. Additionally, blue cones begin to be specified. Blue cones express Dio2, which further increases local thyroid hormone levels. When this is high enough, thyroid hormone begins to signal through THR-beta to promote green-red cone fate rather than blue fate. As green-red cones are specified, they also express Dio2, which reinforces this fate decision later in development. I'm excited to continue working to better understand how thyroid hormone is regulated in the system. Thank you for your time and the opportunity to present this work.